Good afternoon, everyone. Thank you for attending today's talk. It is my pleasure to introduce Dr. Clayton Curtis, a professor of psychology and neuroscience at New York University. And normally I wouldn't mention where a speaker grew up, but today I have to because Dr. Curtis was born and raised and grew up in Dallas, near White Rock Lake, until he ventured off to UT Austin for his undergraduate studies. And he completed his PhD at the University of Minnesota and a postdoctoral fellowship at UC Berkeley. His work focuses on the neural mechanism of spatial cognition, including spatial memory, attention, action planning, and working memory. And by integrating techniques such as fMRI, TMS, MAG, and psychophysics, Dr. Curtis has significantly advanced our understanding of cognitive functions of prefrontal and parietal cortex. And his work has widely published, contributing to both basic science and its potential for applications in neurological disorders. Please join me in welcoming Dr. Clayton Curtis.
All right, well, thanks for inviting me. I really appreciate it. It's fun to come home to where I grew up and see how the city has changed. It's really changed a lot in really good ways too. So I'm happy to be back. And my family's really happy I came back too, because I got to visit with a lot of them. Today I want to kind of talk to you about work that I've been doing very recently. And this is kind of an outline what I hope to cover. So first of all, I'm going to try to tell you about, set the stage. So talk about what's the canonical model that you typically see in textbooks of what is the neural substrates for working memory. And then I'm going to tell you about a challenge to that model. So this challenge to the model was referred to as the sensory recruitment model of working memory, which basically argues that we should modify this canonical model a little bit. And then I'm going to bring even further challenges to that model. So this is based on some work that we have now that's under review, so it's very fresh. So it'd be great to get your feedback. Then finally, I'm going to end with making some claims about how control processes in working memory are really important to consider when we're trying to understand working memory. And a lot of the confusion has been about not really recognizing control processes as much when doing a lot of this work. So I put this little timer up there to remind me, because there are going to be some slides where I'm going to, just for time's sake, gloss over really quickly. I'm going to just hit a couple of points, and if you think you have questions about those, or you think that's interesting and you wish I wouldn't have skipped it, then you can ask me in the question and answer. But just I've practiced the timing a little bit, I just want to make sure that I don't go over. So if you see that, you know I'm going fast. Okay, let's get started. So working memory is I consider it to be like a building block that's necessary for almost all high-level cognition. So including your ability to understand me right now. You're storing in memory for short periods of time things I said before, integrating things I'm saying now so you can understand what I'm talking about. Working memory has two components, two major components. One of them is a storage component. And theoretically, the storage component involves the representation of information. It's held online in some kind of active state, so it can be retrieved easily. One of the important things about working memory that I like to think about is it increases the duration with which information can be processed. So by keeping something in active state, even after it's gone from your environment, this really is the key to why it's really useful for a lot of cognitive processes. Working memory also involves integrating information, transforming information, and it's, that this storage component part is kind of like akin to what we hear as like short-term memory. So short-term memory is like the storage component. Now there's another component as well, and this other component involves these controls. So the controls are basically operations that are performed on the contents of working memory. I'm trying to find my cursor. Is it on? There it is. Huh?
It's thicker.
Yeah. Oh, well, anyway, these operations are thought to, are performed on the contents of your memory. So they can transform a stimulus into a format in such a way that it can be more useful for guiding these memory guided operations. Also to kind of overcome the capacity limitations that you see here. So for example, here's a classic change detection task that's used as a short-term memory measure. Flashing a bunch of colored squares, there's a gap, and then the subject has to indicate whether something changed or not. So you can see this square changed. We know that the storage component is resource limited because as the number of items increase, your performance goes down. This is a hallmark feature of the short-term memory, the storage part. It's capacity limited. The control part is useful too, because one aspect of the control part that's sometimes overlooked is that you can actually allocate more resources to certain items if you think they're going to be relevant. So we've done a lot of work in this. Here's an example where we use what we call our priority pie. It's like a cue that indicates the probability that one of these 4 items that are presented will later be tested. So the red one is it's a big piece of pie. That means about 60% of the time, that's actually going to be the one that's tested. This is 30%, 10%, and 0%. And when we look at a person, then they're later told to make an eye movement to one of those, a memory guided saccade, and we measure the accuracy of that eye movement. This gives us a nice continuous metric for memory performance. It's very sensitive. Love it. When we do this, we've replicated this now, half a dozen times. If this is your probability, you get lower errors if the probability is high. This suggests that subjects are applying more memory resource to the item that's more likely to be tested. So if you think about this, this is a control operation because it's basically trying to overcome the capacity limitations that's in the storage system. So it allows you to kind of optimize your memory and mitigate the limitations of storage. We also have a computational model that tries to model these two terms. So the model basically has a kind of a storage component and a control component. The control component controls the degree of the resources attributed to the different items. And we've used this in a lot of studies now to try to understand how people may optimize their memory performance. Okay, so that basically describes this dual component aspect of working memory. Now, if we think about your neuroscience textbooks and cognitive psychology textbooks, if they talk about the neural substrates, almost invariably the canonical theories of working memory center around this finding that macaques in the macaque prefrontal cortex, specifically the principal sulcus of the macaque PFC, shows persistent spiking during a delay period. So if they're doing a simple memory-guided saccade task, which I'll use a little bit today, you're showing a stimulus, there's a delay period where the animal has to remember the location of the stimulus, and then they're cued to make an eye movement, a memory-guided saccade, to match their eye to their memory. This is done in monkeys a lot, and you can also characterize the receptive fields of the neurons you're making these recordings from. So if you put the stimulus inside of its receptive field, you tend to get persistent activity that's thought to kind of bridge, kind of like a mnemonic bridge over time between the sensory event that disappears and the memory-guided motor response that occurs later. And you can compare that to trials in which you're recording from a receptive field of a neuron that's over here where the target's away from it, and you get no spiking. So there's memory-specific persistent activity. So this is obviously you're looking at this, you're like, wow, this is a really viable mechanism for how we maintain information in memory. And in fact, if you do a lesion to this principal sulcus in the macaque, you also induce memory impairments. So this is the classic thing you see in your textbooks. So people assume this is probably how the human prefrontal cortex supports working memory too. I'm going to just briefly talk about this. if you place a tracer, an injection of a tracer into layer 3 of the lateral prefrontal cortex, you'll see you'll identify collateral branches of connectivity that's quite extensive. And these are excitatory connections. And Patricia Goldman-Kish did some of these anatomical studies with her collaborators and essentially made up a theory for what this means. And so she basically speculated that, well, maybe these branches are the way in which the neurons that are in this column here are influencing their neighboring neurons that have receptive fields that are thought to be similar. So if there's the spatial selectivity for 225 degrees in that neuron, then these neurons tend to have lateral connections to nearby neurons that have similar tuning. And this was basically just inferred from what we knew about V1. So in V1, we know much more now, and we knew much more back in the 80s as well. And we knew that neurons in V1 that are tuned for, let's say, 45-degree orientation tend to be highly connected with neurons that are similarly tuned in orientation. So she just assumed that's probably the same case here. And then her model involved basically the idea that there's kind of recurrent excitation in this network. And this recurrent excitation is a way in which the stimulus input at the beginning of the trial might persist over time. This was formulated later by Xiaoxing Wang into a computational model. And the idea is that this recurrent excitation allows for the persistence of activity. You can see in this population bump, this is actually the representation of the attractor, and it's centered at the location of the stimulus here at 180 degrees. This population vector is pointing towards it. And it also has, and these are each of the neurons that are tiling 360 degrees of visual space. It also has an inhibitory component that helps tune the neurons and suppress activity in parts of the neurons have receptive fields that are away from the stimulus. So this helps stabilize, prevent runaway, and balances the recurrent excitation. So this is all great. This is the canonical model that you see in textbooks. You've probably seen these for quite some time. Now, in 1993, John Janides and Ed Smith did the first PET study, the first neuroimaging study of working memory, and it was a spatial working memory, and they expected to see one blob in the DLPFC. I know this. I talked to them about this. They expected to see just one blob in the DLPFC, and that's not what happened. What happened was they found activation during a subtraction between a task that had a memory and a task that didn't have a memory in parts of the parietal cortex, parts of the precentral sulcus. That's just on the border of prefrontal cortex and frontal cortex, not at the more anterior places that would be predicted by where the location of the principal sulcus might be in the humans. There's no principal sulcus in humans, but we know a little bit about what the likely homologs would be. And there's an absence of memory-related activity in those areas. But we find them in parietal cortex and in this more posterior part. This SPCS means it's the superior branch of the precentral sulcus. And the IPS 2 and 3 and 0 and 1 are retinotopic maps that are in the parietal cortex. Now, I'm assuming that he didn't map retinotopy in the study. I've gone back and relocalized where he found these activations. This is probabilistic. So this was both disappointing, but it also kind of, looking forward, it predicted about 20 years of follow-up studies. So this basic network of frontal and parietal areas has been found in several dozen fMRI papers of working memory, especially spatial working memory. And so, again, we have a slight discrepancy, though, than what you might expect from the monkey. Now, here's an example of what I talked about the last time I was at UT Dallas in 2008 when I gave a talk here. I was basically talking about this data at the time. So I thought I would kind of revisit it here. So again, here's an example of this exact same pattern. and along the precentral sulcus here, you can see in the frontal cortex, and along the interparietal sulcus and the parietal cortex. We find activity doing this exact same memory guidance and CAT test that Funahashi and Pat Gomer-Kish did. So some people at the time were saying, John, Ed, you're just doing the wrong study. You know, you're doing this funny task, this subtraction. So that's why you didn't get the activity that predicted in the DLPFC. So I went and did the study exactly how the monkey did it, and I was able to isolate delay period activity because I have a long delay, and sure enough, I find activity that looks like this. And now we can flash forward to today. We can use much more fancy techniques to look at basically how different cortical areas and subcortical areas, like here, like the superior colliculus, respond during this simple task. The point is the code seems to be really distributed across many cortical and subcortical areas. I don't have it shown you, but we also have found evidence of memory activity in the LGN. And so all of these areas are defined by using a retinotopic procedure that we developed. That's a kind of taking the standard population receptive field method and then adding a twist to it. And the twist was two things. One twist was that we had people, instead of just passively viewing like some kind of bar that's flashing, checkerboard bar that's traversing the retina, we had people have to do a task in that bar that required really careful attention to as the bar moves back and forth. I have slides of that if you want to see it later. We also, the other component is we had to add a nonlinear component, and that was really important because as you move away from V1, V2, and you get into parietal and frontal cortex, the receptive fields of the voxels tend to be very nonlinear. So if you basically only cover a small portion of the receptive field, it almost maximally responds. That's not the same thing in V1. If you cover half of V1, you get half the size of a response as you do if you cover the whole receptive field in V1. And so that's a very linear response. This is a very nonlinear response. So we did those two things, and all of a sudden we see retinotopic maps that start in V1, and like a ribbon, they go all the way up the dorsal stream. So you can see those colors represent the, there's a map here, that's a key. There's the upper visual meridian, the lower visual meridian, and you can see, for example, we start here in IPS 0. We have a map that starts from the upper visual field to the lower visual field, then the lower to the upper, then the upper to the lower, and so forth. So we get basically upright and inverted representations of the contralateral visual field as we traverse up the interparietal sulcus. And then we also find maps that are in the frontal cortex too, 2 or 3 maps in the frontal. I'm going to mainly focus on this one today, but we find other maps as well. And once we do this, then we can ask the same thing that Funahashi asked, which is, okay, what about when the stimulus is in the receptive field of the voxel as opposed to into the neuron? And we can compare those responses to where the receptive field is 180 degrees away from the target, and then that's in the dashed blue lines. And you can see 2 things here. So let's start with V1. So V1 doesn't show the classic persistent activity. It shows a little bump, and then it goes right back down to baseline. This is why a lot of people have considered that V1 is not an area important for working memory. It doesn't show persistent activity. But it does show quite good selectivity between when the target is in its receptive field versus 180 degrees away from it. And then if we start moving up, you start to see 2 patterns that happen, two gradients. One, the amplitude of the delay period activity gets larger as you move up the visual hierarchy here. And it's very large in this precentral sulcus here. The other thing that you see is as you move up the hierarchy, the selectivity tends to get a little bit smaller. I'll show you a quantification of that. So here on the left, this is the amplitude of delay period activity plotted as a kind of going up the visual hierarchy, going from occipital to parietal to frontal cortex. And you can see on average, the amplitude gets larger and larger and larger. And then if you look at the delay period selectivity, this is kind of like the ratio of those two, you can see that it goes down and down and down. So there's not much selectivity once you get into these higher order areas. There's slight selectivity. So that's not what you would kind of expect from this canonical model. And we've tried to understand why that's the case. And one thing we've turned to is using more quantitative ways to ask what kind of information is in these populations. So instead of just looking at the amplitude of the bold time courses over the course of the trial, we can ask what kind of information is encoded in the population of voxels in that region of interest. And what I have plotted here is examples. I will get back to this technique in a little bit because there's some other aspects of it that I want to talk about. But here, this is the target location. This is the true angular position of the target around the clock. And this is the decoded location. This is after we use some fancy decoding algorithms to decode this. And so if you look along, if a perfect decoder would be right along this line here, right, the diagonal. And you can see V1 has a lot of information during the delay period. This is all the last half of the delay period. So in the last half of the delay period, you can decode where someone has a memoranda stored in memory. Yep.
Each dot is a population receptor field.
Each dot is a trial. And this is examples of each dot's a trial. And so we can get decoding on a single trial level using this Bayesian decoding technique. And you can also see the same thing. So it's really good decoding, but you get decoding pretty good. That's everywhere. Even in the precentral sulcus, we can decode pretty well. Now it's not as good as V3 or V3AV or these early areas. This is surprising. So it means despite the absence of persistent activity in these early areas, there's information about the target in the population activity. Just to give you a sense for what it looks like in the middle frontal gyrus would be up here where we see this kind of where the human homologue of the principal sulcus would be. This is at 7T and at really high resolution. Again, we see, you can squint your eyes, you see a little bit along this diagonal. The reason why it's in these columns is we only had like these number of locations, and there was just a little bit of jitter around that. So it looks kind of funny. But again, you can kind of see a little bit, but not much. There's not much information there. And when we do this with a bunch of subjects, we find that maybe 1 out of 5 has significant information. There may be significant information about the target's location, but it's very weak comparatively. Yeah.
So do you have a sense of the dots that are not on the diagonal? Are they a product of errors in the decoding or errors in the actual memory representation?
We think both, because as I'll show you later, they're actually predictive. So when you make an error that's, let's say, clockwise, your decoder, which is from several seconds before, also it has a representation that's a little bit clockwise of true veridical. So on a trial-wise basis, we find this correspondence between the decoder and the behavior. That, I tell you what, that's hard to find in neurophysiology with animals. Okay, so the key thing is, how is it that this textbook area that seems to be the support for working memory, the lateral prefrontal cortex, it doesn't contain much information about the stimulus. It doesn't show persistent activity. I didn't show you this. It doesn't show persistent activity. So today I'm going to try to answer that question. Okay, so that's just my question. Try to answer that question. Okay, so that's the point 1. So point 2 is this rise of the sensory recruitment model of working memory. Now, after, I published this paper when I was a postdoc, and I like one thing that there's certain things you go back to old papers that you really like, and certain things you kind of don't want to read your old papers. This one is one quote that I really like. So this was in a review that we wrote together. It says, I'm just focus on the quote here. These new findings suggest that the DLPFC aids in the maintenance of information by directing attention to internal representations of sensory stimuli and motor plans that are stored in more posterior regions. And so this is a reformalization of what the role of the prefrontal cortex may be doing in working memory. It's not storing information. It's aiding somehow in the maintenance by some type of top-down control. And you might say, okay, well, of course, that's what everyone talks about. You know, lateral prefrontal cortex and control, top-down control, that's what it does. But try open, go to your, I saw, Nick, you have the cognitive neurosciences textbook. Go look at it. It doesn't say that. A lot of the textbooks that we're still teaching today talks about the standard canonical model where you see persistent activity in the frontal cortex, and this persistent activity is carrying the information that you have in working memory. It doesn't say that it's a control signal that's targeting a posterior area where the information is stored. It does not say that. So we speculated about this. We didn't have the data to test it, and I didn't really know how to test it at the time. This was in 2003. I didn't really know. So I was really jealous when I actually reviewed this Harrison Tong Nature paper 2009, and I got it, and I was like, oh, damn, I should have done this paper. I should have done this study. Frank knows I was reviewing. And so it was simple. John Sorensis also did it as well. They published it almost at the same time, and both of them published in really great journals. But the idea is simply, I'm going to go fast here, is if you're shown a grading and asked to remember it, you can actually decode the contents of that grading from a memory delay in early visual cortex. This was shocking to a lot of people because at the time, very little attention was being paid to the role of visual cortex in working memory. It was all about frontal and parietal cortex, everything. It was like a myopic kind of machine that was focused mainly just on prefrontal cortex. And these results finally appeared, and they supported what a lot of people were kind of starting to wonder about at that time. Hey, wait. I think this might be more of a control thing than a storage thing. And so this was the first evidence, and it basically opened up the floodgates. There has been now probably, I don't know, at least 24, maybe 3 or 4 dozen imaging papers that have replicated this result, showing that you can decode the contents of working memory from V1. Any kind of visual feature. You name a visual feature, it can be decoded from V1. And usually better than other areas. That's surprising. Okay, so what do we make of this? Well, it suggests that this new way of thinking about the sensory cortices and working memory. So the idea is essentially that there are sensory mechanisms. Think about this giant manifold of visual cortex that's used for converting everything we see into internal representations of all these different features. It's basically going to use that encoding manifold to store information. So how does it store information? That's the big question, because V1 doesn't have a lot of the properties that the prefrontal cortex has that actually makes the prefrontal cortex really good for memory, but V1 really bad for memory. And to a certain extent, we don't want things lingering around on our retina. We don't want things lingering around that just appear to our retina because that can interfere with new incoming information. So these are the big open questions that people are kind of fighting about now. So this led to this idea of a sensory recruitment model. Working memory is recruiting sensory cortices for maintenance. And it was basically pretty much agnostic about how. It was just, it does. The early formulations of it didn't even say anything about the prefrontal cortex, really. It was just, we used early sensory cortex somehow to maintain information. So in our hands, we're trying to kind of understand how this might work. And so, for example, we and other people have asked this question, well, if it's using these mechanisms for perception, then how similar are these representations, sorry, Mick, I used the R word, these representations during perception to during memory? How similar are they? Are they interchangeable? And we've done a bunch of studies on that. I'm gonna talk to you here about one that was a little bit different. It's basically asking, the visual system has been, studied more than any other part of the brain. Maybe the hippocampus and the early visual cortex compete for which has been studied the most. But one thing we know about the visual system is that it uses a probabilistic population code. So kind of an Bayesian kind of framework. And according to this hypothesis, if working memory also uses this probabilistic population code, then we might expect that that a population of neurons not only represents the feature of the remembered target, for example, like its location, it stores it as, or it represents it as a probability distribution over the stimulus feature. In this case, it could be the polar angle of the target. And what's interesting about this is it makes some really powerful predictions. One prediction is that the mean of this probability distribution, if you could estimate it, would essentially relate to what your memory is. And the uncertainty of your memory would be related to something like the standard deviation of this distribution. So I'll tell you about the task in a second, but you can imagine this probability distribution, and here's the location of the stimulus, and the mean of this would be basically related to your memory report. What is it that you have in your memory? And the standard deviation of this distribution would be related to how certain you are about your report. So we can actually build a Bayesian decoder that can estimate this full probability, it's a likelihood function. And we can use that to make estimates on single trials about these two quantities. So the way we tried to relate this to behavior, because as I was saying, I really want, I believe things when they actually match behaviorally well. You can get a lot of hocus pocus stuff from decoding stuff in the brain, but when it actually corresponds in a way with behavior, I start to believe it. So the task is simple. There's a dot that appears. You remember its location for a long delay, and then you make a memory guided eye movement to it, and then an arc appears. And you turn a knob that says, okay, how confident am I in memory? Basically, how sure am I that I'm within at least this error bar? And you're playing for points, and people love this. We didn't even pay the money. It was like a video game. They were like trying to get as many points. People were competing. So it was fun. It actually was kind of fun. So obviously the smaller the bar, the more points you get. But if you made the bar too small, sometimes the dot would appear just outside of it, 0 points. And that was really disappointing. We measured people's eye movements. We found really good correlations between their memory error and this arc length. So you can think of this as the better accurate their memory was, the more confident they were in their memories. So these are things you might predict behaviorally. And then our decoder essentially is trying to use these basis functions that are tiling visual space to make a trial-by-trial estimate of what someone has in memory. And so in a population of voxels in an area, we do this for every voxel. Every voxel is modeled by this linear combination of these basis functions. So for example, in this one, you would expect that the target might be somewhere that's centered around here, but this one's a little higher, so maybe it's a little bit to the right. Okay, so each voxel on each trial is modeled by this. So you have another trial, the same voxel, where the target's more closer to this location, another trial where the voxel is closer, the target's closer to this location. And overall, over time, you can get these estimates where you average over not only just voxel, but not over voxels for a single trial. And this could be one trial, this is one trial, and this one trial. And then we actually take the mean of this, and that would be what we're calling what you remembered. And then we're taking the standard deviation, and that's what we're calling your uncertainty. We get these two quantities on each trial, so we can do it for this trial, and this trial. And then essentially what we can do, I've already shown this before, we can take the target location, the real target location, and plot it against the decoded target location, these means. And we find in V1 really nice, V3 is always spectacular. Early parietal is mid-parietal cortex and even frontal cortex. We find representations of the target location encoded in this population activity. Here's an example of the Dakota error. So the error is it's a little bit worse in frontal and parietal than it is in these visual areas. And we can also ask how does this correspond to behavior? So here's your memory errors, and here's your decoding error. And we find these kind of linear relationships where when your decoding error is a little bit counterclockwise, your memory error is also a little bit counterclockwise. And we find correlations, not in V1, but in this mid-visual area and in frontal and parietal cortex. There's some relationship there. And that's nice to see. And then if we do the same thing for uncertainty, here, the decoded uncertainty is this standard deviation of each of these, and this is the reported arc length. Again, this is trial-wise. We find, again, in at least V3AB and IPS, we find these correlations showing that as your memory is more uncertain, the decoder is more uncertain. Okay? And this is not like wild guess or forgetting. The people are not making, I mean, if you look at the arc length, these are tiny, tiny degrees. Memory error, this is all basically less than 2 degrees of memory error. People are not forgetting. This is not like lapses of attention or they're falling asleep or anything like that. These are, we exclude all those trials. We only focus on good behavior, and we find these results. So this is exciting because basically working memory and uncertainty seem to be kind of multiplexed in the same population. This is consistent with this concept that's been really powerful for explaining early visual cortex in monkey, rodent physiology, and a little bit of human psychophysics and neuroimaging work. And this is explicitly during this delay period where nothing is happening. They're looking at a fixation point. And the key thing for us that's really compelling is that these features that we can decode are actually related to behavior on a trial-by-trial basis. I mean, most of the correlations with behavior that are out there are subject level. So you'll see, okay, this person has a better decoding, and you find a correlation between the subjects with good and bad decoding, with good and bad behavior. But to see it within a subject and with the direction of the kind of mistakes that you're making, I think it's much more stronger evidence that these decoded values are important because they're tapping into behavior. Okay, so not everyone buys this, which is fine. So many kind of believe that V1 is just not necessary for working memory, and they believe that mainly because they think that the neuroimaging evidence is too indirect to be compelling. The monkey physiology is a little bit mixed in support of this idea. And so some people kind of think that the activity that you're measuring in V1 is not really V1. It's the exhaust of some kind of computations that are performed elsewhere. And maybe V1 inherits that, but it's not necessary. So no one's ever been able to lesion V1 and do a working memory study because you couldn't see. And so what we did is we did TMS. We did a really careful TMS, and this was a kind of a important study to do because the sensory recruitment model is basically on the stand. And if there was evidence that V1 did not cause any impact on working memory, it would be very damning to about 15 years of neuroimaging work. It would be consistent with, you're just measuring the readout from the other areas that are important, but V1 does not matter. So let's get back to the PSC and stop talking about this nonsense in V1. So what we did was, if you do retinopathy, you have to ask yourself, where do you have access? Where can you access the TMS? And we can really only access this dorsal part, which is in the green and the cyan here. That's the part that's accessible with TMS. And here is the population receptive field coverage of that area. So you would expect that things in the lower contralateral visual field would be accessible. And in fact, when we do TMS to this area and we have people draw where they see phosphines, phosphines are visual aberrations that you can invoke with TMS to V1. It looks like little staticky, flashy lights. It's unnerving, actually, to have that, to see that static. But anyway, we have subjects draw where they see it on a bunch of trials, and we define this as what we're calling the phosphine field, where we know that V1 is impacted by the TMS. We do modeling of the electric field, and we can define V1 here in both of these hemispheres. We can plot what part of V1 is being impacted by TMS, and this is in volts per meters. And we can show that the part of the V1 that represents where the phosphine field has a lot more voltage than the part that's opposite. Okay. So now the question is, what happens when we do an experiment? The experiment is, again, we're working with the same memory-guided saccade tasks for a variety of reasons. One, that memory-guided saccade is exquisitely sensitive. So we can measure, if there were small perturbations down to about 1/10 of a degree, we could still capture them with our eye tracker. We've been doing eye tracking for 25 years in my lab, and we have really good ways to make really super precise measurements. So in the no TMS, you present a target, there's a delay of 4 seconds, they make a response, and we just measure the error. You could do this in the left visual field, right visual field. These are places that are the mirror image. Here, the mirror image of targets in the phosphine field. So here's the phosphine field. We put a target somewhere in it. It could be anywhere in this phosphine field. They have to remember it. And then either in the middle or the early delay, we apply a triple pulse of TMS. That's been titrated down to where people do not see phosphines anymore. And then they make an eye movement. This is the control. This is a control, no TMS control. Here's another control. Here's where we put a target outside the phosphine field. This is a really rigorous control. But you're still applying TMS to visual cortex. And as you saw, there's still even a little bit that gets over here into the other V1, but it's small. So here's the data. So here we're comparing here out versus in. And let's start the no TMS. So the no TMS, on average, they make about accuracy about 1.5 degrees of visual angle. So that's basically maybe your finger extended out, the width of that. It's their average error. And let's focus on the middle TMS. This is at 2 seconds into the delay period. You can see when the target is in the phosphine field, you get an increase in errors of about 0.3 to 0.4 degrees of visual angle. And then when the target is in the away out of the phosphine field, it's basically essentially the same as no TMS. Yep.
May want to talk about this later. I'm sure reviewers brought this up. That data are beautiful, but you could argue that the effect is indirect.
Yeah.
Stimulating V1 is sending information further up the system, disrupting bits of the system that are really doing the working memory storage.
Yeah.
So it still doesn't prove that V1 is the site of the storage of the information.
I think that is that leaves a little uncertainty. However, luckily for us, there has been work done on this, and there's been now animal models that are studying TMS. There's also been combinational TMS and fMRI and PET studies trying to show. And I was kind of surprised about this, but it appears as though the only thing that could possibly be affected with things that are monosynaptically connected. And so you can say, well, okay, what about this V1? Is V1 monosynaptically connected to the GLPFC? No, it's not. Is it to the frontal eye field, which is likely to be part of the superior precentral sulcus, that dorsal map in the frontal cortex? There are a very small number of monosynaptic connections. I mean, they are tiny, but they're there. Could they account for this effect? I don't think so. Also, yeah, I don't know. That is something that the reviewers brought up, and we did discover this great literature. That was very helpful. I'm going to send thank you. I'm going to send you thank you emails to the authors sometime. So yeah, it's just, there's no other way to do this work though I can think of. Okay, I mean, even if you're doing like, you know, microstimulation, Muskomal, in animal models, you still have this problem of diaschesis, right? It's always a problem. You just, you know, it's what we have. But yeah, you're right. It's something we should worry about. In the early delay, we get the same thing. We had this as our control because some studies have shown that if you do TMS right after the stimulus, you can sometimes impair working memory a little bit. And they argued this was involved in the recoding the information from a visual input into something that's like more memory. I don't know if I believe it, but it's interesting. Okay, so this suggests that V1 is important. It is something we should pay attention to. So one, just as like an interim summary here, the content of working memory is decodable from these neural patterns of activity in visual cortex, including V1. It's predictive of these memory-guided behaviors on a trial-wise basis. And we now know that V1 is, we think is necessary for precise working memory. We're not claiming that it would like erase someone's memory, like say for a face. We think it's something about what's V1 good for. Okay, so things like orientation and motion and contrast, like these real low-level visual properties like location. We think that there's some matching between what an area is encoding and how it might be used for memory. We're trying to test that later. Now it has some problems here. One, how can V1 store working memory while simultaneously encoding incoming perceptions? People speculate that, well, maybe it's a laminar thing. So, you know, the memories in these superficial layers and the incoming still in the middle layers. No one knows, but that's one speculation. And I want to answer the second question of how robust are these working memory representations in visual cortex to distraction. And so there had been a couple of other high-profile papers, Nature Neuroscience papers, where they come up with disagreements about their findings, and we were trying to make sense of this. Why someone finds that V1, when you have a distractor, just loses all ability to decode anything. Another group finds that's not the case, but it kind of depends on the type of distractor. And so we were looking at this saying, you know what, there's some problems with the way they've done things. The biggest problem was that they didn't have any behavioral effects at all. So they're doing distraction, but it's not distracting. So that was a big problem. So we decided to do something where there is some distraction. So again, we turned to our favorite memory-guided saccade task, our workhorse in the lab. And we present a dot during the delay period. There's like a little cloud of dots, and they're swirling either clockwise or counterclockwise, and they have low coherence. And the subject has to make a decision about which direction it's swirling. And so now the distractor is relevant. In those previous studies, it was irrelevant. You could ignore the distractors. They were completely task irrelevant. And they were during the entire delay period. It was just like, you know, showing pictures of gabors and gazebos and all this stuff during the delay had no impact on behavior at all. And the effect it had on these representations in early visual cortex were very mixed. So we have this, and we show that there's nice effects on the behavior of the subjects, but it's not catastrophic. We didn't want any kind of catastrophic interference because what would we be decoding at that point? We wanted to see if it was being nudged around. So here's some of the time courses from the task, and you can see in these early areas and these later parietal and frontal areas that you can see in the red. This is no distraction, and again, I'm showing you the data when the target is in the receptive field. You can see this little blip that happens every time there's a distraction. This distraction and this little blip gets bigger as you move up this visual hierarchy, as does the persistent activity. Again, it's another demonstration of this effect. Here's the distractor. So this was when the distractor is in the receptive field versus no distractor. And again, you get these big responses that are time locked to the distractor. So cool, kind of a sandy check. It's kind of what you'd expect from the bold time courses. But what can this tell us about whether or not the information was lost after the distraction?
Is in that group
Yeah.
fight, is the delay related activity in V1 smaller than you normally see for the distractor absent?
No, that's about it. That's like classic. We've seen this multiple times. There are a couple of exceptions 10, 15 years ago that we had, but yeah. So I'm gonna go through this really quick because I just think it's super cool. This is a single trial in which we use a encoding model, or we invert an encoding model to reconstruct the representation. This is over time. These are 750 millisecond TRs. This is where the target is at time 0. And then you can see there's pretty much activity stays highest along the target, single trial. And then here's where there's a target, and here's the distractor. So you see you get it's highest right aligned with the target. Distractor comes on, it wiggles over here, but then it comes back. And we see this in many subjects. You can see here's all the different locations of the congruence between the target and the distractor. Obviously, when they're aligned, really close to each other, you get big activity. But they kind of wiggles, but comes back. And every single time, it comes back. And that's basically the finding that we had. Essentially, this is now group average, no distractor. Here's the distractor. You can see that here in V3AB. Nice tuned representation through the delay period, and then it kind of dips, and then it comes back before the end of the delay. And we can confirm this with a statistical procedure, which is basically estimating the fidelity. You can think about it. What's the amount of information that's consistent with the item being at the location that it was queued? And so, again, the key thing is in the blue. And the blue is the distractor trials, and you see this little, when the distractor comes on, a little dip. There's a dip, but it always comes back. There's never a full loss of the information in any of these areas, V1, any of the areas that we've studied. And so there is some resistance to the distractor in these populations, even when the target, even when the distraction is something you have to behaviorally pay attention to and make a decision about. So we think that there's the resistance probably comes from there's so many brain areas that are all representing this information, and so there's a little bit of the redundancy builds in some distractor resistance, but it's also related to behavior. So if we look at, here's our memory error that they make, and if you look at the neural bias, these are a neural bias is basically our measure of the degree to which the distractor caused the representation to go one way or the other. And you can see that when the distractor, when our decoder says something's gone a little counterclockwise, we find that memory errors are again kind of counterclockwise. And when the memory errors are clockwise, we find that the decoder goes a little bit clockwise. And so the distractor, we think, and these are, this is distractor only trials because it's the bias. We think that the distractor is pulling the representation a little bit towards it. So even though I said it's distractor resistant, it's not immune to being biased by the distractor. And so those earlier findings, these basically suggest that it's just more of a nuance effect of distraction. Causes this brief dip in the fidelity of the memoranda. And also, we find that in the same population, we get joint encoding of the distractor in the memoranda's position. Again, we get these little small biases in memory errors, which can be predicted by the biases in our neurodecoding. And we find this selectively in early visual cortex. Okay. Now, getting back to this, we know that as you increase load and do neuroimaging, you get greater effects in prefrontal cortex. So load 3, load 6. This is from Bart Ripma. It's not here. Disappointed. And you also find it in parietal cortex too. And these curves that you find in parietal cortex kind of map the curves that you see when someone's trying to estimate capacity. So as your capacity kind of plateaus, so does the bold signal in these areas plateau. So this suggests that the frontal and parietal cortex act as some kind of a capacity limited storage system. That's what one interpretation of this is. Now, but we asked this additional question: how are resources allocated in a strategically controlled manner? Because we think that that could account for some of this data. So the way we did this is I'm wrapping up soon. This is a study in which we had a priority task. So they were told that one of these two items was going to be tested twice as often as the other. We do the same delay period to make a SICAD. We get nice behavioral data showing that their errors are lower for high than low. Again, all this is sanity check. Just here's what the decoding looks like for the high priority trials. Here's what it looks like for the low priority trials and stuff. It looks like for everybody. It's just beautiful. You can just tell this is a noisier representation. And so we asked, we asked, well, if we estimate the gain of the response in the high priority trial, high priority target, and at the same time on the same trial estimate the gain of the response in the low priority trial and create a ratio between those two, that can be our estimate of how much you're prioritizing one over the other neurally. We can also, we can correlate that with behavioral prioritization, which is basically has to do with these errors. How much are your errors smaller for low than high priority? And we get these correlations in early visual and mid visual cortex, suggesting that there's a relationship. Okay, that's cool. But here what we did next was we took this ratio. So you take a trial one, you get the ratio, store it. Trial two, get the ratio, store it. You have a vector of all the trials, and you take that vector and you correlate it with all the delay period activity in every voxel in the brain. What's the amplitude of persistent activity? And when we do that, we're just going to focus on the red here. We find that in the superior precentral sulcus, we find that the activity correlates. So what this means is in the superior precentral sulcus, when delay period activity is high, it means the subject is prioritizing more the high priority target than the low priority target. So we reason that perhaps what role this superior precentral sulcus is playing is on the control of your resource allocation. And so to test that really quick, we just went to TMS. We TMS this region, and we basically, in the magenta, there's no TMS in the black. In the magenta, the curve flattens out, suggesting that less resources devoted towards more resources devoted towards the low priority trial. So it's like you're flattening out. We can't really get this to go much lower. So you would expect a full kind of crossover, but we don't find that. And there's just a floor effect. Here's single item working memory, and no one can get better than like about 1.4, 1.5 degrees of visual angle. It's just, there's sensory motor noise. So we applied our model to this, and just the take home is here. So if you're just doing the experimental priority, you'd say 2 to 1. This is how much resource you devote to high to low. With no TMS, we get 60-40. This is not the same, and we've replicated this many times. You can ask about it later. But if you expect no priority, complete flattening, be 50-50. And what we find is just a reduction. So if you compare this to this, you can see that we've had a reduced amount of the parameter that represents that prioritization based on their behavioral data change in the high priority trials, and an increase in the low priority trials, consistent with perturbation of this area unbalances that. I'm going to skip this, but we have we can think a lot of old data that we publish now, given these new findings, and we can reinterpret them. I think some of the findings that we published from patients who have resections because of like tumors and things like that, and other TMS studies we've done, now I think differently about those results, and I think we may have been actually perturbing the control regions and not the storage regions. I wish I could rewrite those papers. So in summary, wow, this is looking like a gender reveal. I did not. Okay, now going back, we find strong evidence of persistent activity in these control areas. We find poor decoding though. We find the gain of persistent activity in these control areas predicts the quality of these memory representations in visual cortex. Predicts the strength of working memory resource allocation during prioritization because the TMS to SPCS disrupts its allocation. And we find large responses to distractors in these control areas. And we think we have some new studies going on. We're trying to get at how control might be used to overcome the distractor, protect the contents of memory for distractor, but those are ongoing now. And then in the storage area, find little if any evidence of persistent activity in these areas. The signature of working memory is kind of absent in these areas, and that's why I think confused a lot of people. We can decode the contents, on the other hand, very well from these areas, best. And we can decode different aspects of the quality, which are important. It's not just, yes, it's there, or no, it's not there. We can decode the accuracy, certainty, these properties, they think they're important and relate to their mechanisms. TMS disrupts working memory when applied to primary visual cortex in a very retinotopically specific manner. And when we decode these working memory parameters from visual cortex, they predict memory-guided behavior on a trial-wise basis. The only other big thing is we think that these effects predict how well resources are allocated. They're the effects. And then I just want to thank my lab who did all this work. I just get to talk about their amazing work and represent them. And they're fantastic. Thank you. Yeah.
So a lot of the work you showed was based on relatively brief exposure to past design. The importance of B1 seems to be really strong with the evidence that you shared here, but I was curious if you overtrained with every primate setting. Humans, do you think the importance of B1 to that you described, you all work the same resource allocation?
Yeah, that's a really great question. One of the common suspects of the disagreement between the human and the primate work is the primates often undergo like at least 3, if not 6, 7 months of training before they start recording. And when they're recording, they're getting on the order of 20 to 30,000 trials often. In the humans, that's not the case. However, there are a couple of humans in our studies that have done many trials. So some of them are really well trained. I have been in some of these studies, and I have done memory-guided cicads since the 90s, and I may not have done as many as monkeys have done, but I've done on the order of 20 or 30,000 in my lifetime. And sometimes I get in the scanner just to test your idea, just to make sure. And so far, it hasn't made that much of a difference, except for it makes some of the other subjects look good. I mean, look bad because my data looks so good, and I can't be in these studies anymore. I've just practiced. Yeah, that's a good question. There's been experiments too where people have practiced subjects over months and months and months and measured them over and over again to see what effect learning has. And there are some slight changes, but so far I wouldn't say that they're as compelling as I would have expected, to be honest.
I guess you must have had this question many times, but it's my impression that lateral frontal cortex, classic lateral frontal cortex, inferior frontal, superior frontal gyrus, you tend to see that in working memory tasks in humans. When the memoranda are semantically encoded. So, for example, nonsense syllables or words or whatever, that seems to be the situation where you get really clear activation of these more anterior lateral frontal regions. Do you think your work is fantastic, but it's all on semantically uninterpretable, very low level visual features? To what degree do you think it generalizes to typical working memory situation for a human? Humans don't go around worrying about locations of dots or angles of lines. They worry about whether they can remember what their shopping list is that they read 30 seconds ago. So To what degree do you think there is generalization?
So in some ways, the work that I've been doing is trying to show that the original work that was done in primates, I was trying to build a bridge to that work, and that's why I'm using these simple things.
Okay.
And part of the goal has been to influence the theory that has come from studies to start to reflect something more like what you said. Because we have to remember these are animal models of human cognition. And my battle has been to try to convince them that there's something wrong with the way that their theories are being operationalized. And often some of the neurophysiologists that they are my friends, we argue, but some of them are some of my best friends. We have spirited conversations about this. And part of me is trying to change the way they're thinking about working memory. And I think something has to change. So what has to change? Some people say the opposite of you. They say, well, your task is too simple. That's why you don't see working memory, delayed period activity, or representations in lateral frontal cortex. You're using too simple. But I'm using the same tasks that they used to develop all these theories, and that was the only way to really get traction in that field. They wanted to see the studies done exactly the way they do them. And once I have been doing that, they pay attention more now. Now, with what you said about the activation in the prefrontal cortex in different types of stimuli, more complex stimuli, that's probably true. But even if you keep the item load to 1, you do not see really persistent activity. You can't really do decoding of more than 1 item because no one has a decoder that works for more than 1. We have 1 for 2. That's the first one out there. You can't do that. So essentially, you can't test these ideas yet. But I think what you think is right. I think it's basically the thing that might need to change is them to get over this idea that it's storage in the prefrontal cortex of simple features. I still think it's a control signal, and whether it has to do with something that's semantically encodable, I am agnostic about that. I still think that it probably would still not be necessary. But load, if you increase the load, you do get increased activity in lateral frontal cortex. Originally, that was thought to be, oh, now you've upped the storage demands and now you see it because fMRI is a crappy tool. We all know that. So you need to really up the load to be able to get it to measure something that's subtle. I don't think it's.
I don't mean to prolong. I know we're running over, but just to say, I wasn't implying that the lateral frontal cortex is storing semantic representations, but I think it's controlling the sculpting representation that is maintained posteriorly.
I like that. Yeah, I think that exact thing is true. But yeah, we're turning a corner now in my lab. So if you look at the early stuff I was talking about, it was very simple. Now we're getting into prioritization, and that's kind of like it's okay for me to ratchet up a little bit in that comfort zone. But some of the tools that we use just do not work when you're using more complex stimuli. So I don't know what I would measure. I don't know. Yes.
Maybe so is there a way for you to show the control in terms of the interactions in those units?
Yeah.
It implies that there are direct interactions, something in this interaction that has to bear evidence for the way that it might be.
Yeah, so I showed you one piece of evidence, but we actually have 4. We've had a student that went back to 4, now 5 other data sets that we've collected in the past. And she has done the decoding and got the trial-wise error and in early visual cortex and predicted the delayed period activity of all the brain. And she is found in all 4, I know 4, 5 still being analyzed. All 4 of those, we find that the precentral sulcus predicts the decoding quality. So When it's high, better decoding quality. When it's low, worse quality, less prioritization. So that to me is really consistent with a control operation. And because we're correlating something that's about fold amplitude with something that's not amplitude, that helps a lot, because amplitude correlations, as they go up in networks, and it becomes hard to interpret how that might be playing a role in control, right? So we're kind of happy about those results so far.
Is there any online?
Is there any question from remote audience, Shelby?
Yep, we do have one question. How does neural coding of working memory relate to EEG brainwave patterns?
Yeah, that's a good question. I was shocked when I guess the first evidence for these kind of methods working with EEG started to appear around 2016, 17. I was surprised that you could use the same methods we're using for decoding an fMRI to take EEG data and decode location, orientation. I think now someone did it with color recently. That's surprising to me, but it works. And the one thing to note is look how much data they need though. So in these EEG studies, it's a ridiculous amount of data. Probably you need three hours minimum to get enough data to have these decoders work. It's just a little bit noisier of a measure than fMRI in certain circumstances. I want to be careful here.
Okay, we will conclude today's talk here. Thank you to Dr. Curtis for